Cellular Forms is the latest stage in a body of work that I've been developing over a number of years called morphogenetic creations. Inspired by Alan Turing, Ernst Haeckel and Darcy Thompson, this work looks at the relationship between growth and form. The aim in Cellular Forms was to create a richly emergent system for generating very complex organic structures based on morphogenetic processes. The system uses a simplified model of growth by cellular division, taking inspiration from biology but not trying to emulate any specific organism, and trying to be computationally efficient enough to be run with many millions of cells to create the level of detail that I believe can be biologically convincing rather than just mathematically organised. The project also has aesthetic goals, both for myself and the viewer. I'm interested in exploring the emergent nature of form created by growth systems, and results that may be challenging rather than conventionally beautiful. What unexpected but aesthetically strong results can be achieved? The process is one of emergent exploration rather than directorial control. I'm also interested in the reactions of viewers. I believe there is something deep in human psychology that results in strong reactions to complex organic things. From reactions to the sublime, such as moments of epiphany when viewing the beauty of nature, to disgust and revulsion when faced with disease and decay. What happens when forms are presented in a more abstract manner, not designed to represent any one particular thing in nature, but still retain an almost overwhelming degree of organic complexity? The system I'm using for Cellular Forms models cells as simple particles that are linked to a set of neighbours. Each simulation starts with the same ball of cells. The system contains rules for when particles are selected for splitting, which are based on accumulated levels of nutrient. When the nutrient level in a cell exceeds a given threshold, it splits, and both the parent and daughter cells are reconnected back to the neighbours. There are forces between the linked cells which create dynamic tensions in the structure, creating motion as it develops over time. By altering the rules for generating nutrient, how cells split, and the forces between cells, a remarkably wide range of different organic structures can be created. One additional force that was added was one that repels cells that are in close proximity but not directly connected. This adds an important constraint that helps induce structural coherency. The presentation is kept deliberately simple, the aim being to show the generated forms with as few biases as possible. I use a number of methods to turn the data into visible artifacts, with each technique highlighting different aspects of the generated forms. Structures are either rendered using an external representation, where cells are shown using spherical primitives that are illuminated with a perfectly diffuse light from all directions, or by using a density accumulation X-ray technique that reveals internal structures that aren't apparent in the surface renders. As well as animations, I also create very high-resolution still images of the final forms. These allow an appreciation of detail in the structures that isn't always apparent when viewing moving images. By contrast, the animations from the simulations reveal dynamic behaviour and what appear to be different phases in the development of the structures as they are grown. A number of emergent behaviours are seen to come from the simulations. These include complex structured folding and coherent pulses of cell growth in waves across the surfaces. Variation in how nutrient is created and how fast it diffuses from one cell to another can also have dramatic effects on the nature of the generated structures. In particular, when nutrient is generated by light, if that light is restricted to only come from above, much more plant-like structures are generated, with the forms bifurcating into complex branches. One of the main things the work tries to explore is why complex organic forms can create such a visceral range of reactions in viewers. Even though all that is being presented are typically arrangements of simple spheres, responses range from comments such as mesmerically beautiful through to terrifying or even disturbing. As I have said, the aim with Cellular Forms wasn't to directly emulate any specific biological structures, but to create a system with rich emergence. A wide variety of structures are seen, but it is interesting that all appear to have a strong sense of the familiar, with echoes of organic structures that we are commonly used to seeing. This raises questions about the available range of structures that organisms created by processes of growth can take. What constraints as well as opportunities for complex structure does the process of growth impose?